As I get started, it's with my great pleasure to introduce Sadie Wignall from Northwestern. Sadie and I go back 26 years. I was the second postdoc in the HEAL lab, and she was a grad student at the same time. So we overlapped starting in August of 2000, right? So one of the things I liked about Sadie is she was very thoughtful, insightful, but she's always has a lot of fun. And I know she enjoyed a lot of Rebecca's all the margarita parties we had. But Sadie's a lot of fun, and I've enjoyed knowing her as a an amazing scientist and also as a person. She then went to Anne Villeneuve's lab at Stanford, and after that she came back to the Midwest, and now she's been at Northwestern for several years and is a professor, full professor. And she's going to tell you about her story. But one thing I do know that she's also won since then is because she's just an amazing person. She just won a teaching award at Northwestern. So congratulations to that. That's a great honor. So without further ado, take it away.
Thank you, Anna. So yeah, as Anna said, I've known her for a long time. And so it's really fun to kind of come full circle and to come to Madison. I really appreciate you hosting me today. And we have margarita parties at Northwestern. I've continued on Rebecca's tradition. So my lab hosts a yearly margarita party every year. And we squeeze limes for about 4 hours every July, so it's really fun. But anyway, I'm really excited to be here to tell you about the work in my lab. And so in my lab, we're really interested in understanding how cells divide. And this is actually, I got interested in this as a grad student in Rebecca Heald's lab, and then continuing on into my postdoc, and then also into my lab today. And so I like to start talks like this with these beautiful old drawings from Walter Fleming from 1882, showing here's an interphase nucleus with the chromosomes condensing, preparing to divide. Then chromosomes are lined up in the center of the cell, pulled apart in anaphase, and then the cell pinches into in telophase, creating two daughter cells with the same amount of genetic material. And I got really fascinated with this process and specifically interested in understanding the machine that segregates the chromosomes during cell division called the spindle. So this is a C. elegans spindle in the one cell stage embryo. In green are microtubules, which are dynamic polymers that grow and shrink and capture the chromosomes. They then line the chromosomes up in the center of the spindle and pull them apart. And in most cell types, structures called centrosomes help to build the spindle. So a centrosome consists of a pair of centrioles, which are these microtubule-based barrel-like structures that then recruit something called the pericentriolar material, and it's the PCM that actually nucleates the microtubules of the spindle. So in normal cell division and mitosis, you have a centrosome that duplicates and then moves apart, nucleating microtubules and setting up the two poles of the spindle. And microtubules are polar polymers, so they have plus ends and minus ends. The plus ends emanate away from the centrosomes and then capture the chromosomes, forming what these kinetochore attachments. So they attach to the chromosomes at sites called kinetochores, and then the chromosomes can move to the metaphase plate. And so two kind of like textbook features of this cell division process then is that these centriole containing centrosomes are acting as these cues, these structural organization centers to define and organize the two spindle poles. And also that these end-on kinetochore attachments are really important in driving the congression process of chromosomes to the middle of the spindle and also segregation. But my lab now studies a different type of cell division, a specialized type of cell division, that of oocyte meiosis. And this oocyte meiosis is really unique for a few reasons. First, because oocytes actually lack centrosomes. So oocytes degrade their centrosomes. So this is now a C. elegans oocyte, and this is the oocyte spindle here. And there are no centrosomes present, so this spindle over here has to form these two poles, create this bipolar structure in the absence of centrosomes. And then what happens is actually when this is actually the sperm DNA that has come in to fertilize this oocyte, it brings in associated centrioles. Now, during the meiotic divisions, the oocyte divisions over here, these centrioles aren't doing anything. So they don't have the pericentriolar material, so they're not nucleating microtubules. But later, once these divisions are complete, they'll become fully functioning centrosomes, and they'll help facilitate the mitotic divisions of the embryo. So since these divisions really happen in the absence of centrosomes, there are unique mechanisms. And so my lab's really interested in understanding kind of the differences that occur in mitosis when there are these centrosomes. These microtubules are nucleated from these pole structures compared to oocyte meiosis where these are lacking. And so we asked two kind of related questions. First is, if you don't have centrosomes, how are you even forming this barrel-like, kind of American football-shaped spindle structure with two poles? And second, how are these spindles acting to mediate chromosome congression and segregation? And since I showed you pretty diagrams of mitosis on one of my first slides, I also like to show this. So oocyte meiosis has also been studied for a long time. So these are drawings by Theodore Bovary in the 1800s of oocyte spindles in a parasitic nematode called Ascaris. So I don't study Ascaris, but we use a related nematode, C. elegans, to study these questions of oocyte meiosis. And so C. elegans is an awesome model. As I know many of you in the audience know, there are a lot of great worm labs here. First, they're genetically tractable. We can do screens. There's potential for gene discovery. We can now use CRISPR to tag proteins, make specific mutations. And then also they're transparent, and that means they're amenable to live imaging. So we can express proteins, GFP tagged proteins, and actually watch the meiotic divisions in live animals. And this makes C. elegans one of the most experimentally tractable model organisms that have oocytes. So of course, meiosis is studied across many organisms, yeast, all kinds of different organisms, but C. elegans is one of the simplest that has both oocytes and sperm, and you can study these specialized cell divisions of the oocyte. So because I'm going to be showing you lots of diagrams and pictures of C. elegans chromosomes, just want to walk you quickly through what these chromosomes look like in meiosis, because it is a little more complicated than mitosis. So because this is meiosis, pairs of homologous chromosomes, as depicted here, have to come together and line up and pair and then recombine. So there's recombination events that happen to designate these crossover events here. So in C. elegans, there's only one crossover per chromosome pair. And then these chromosomes reorganize around this crossover. So the crossovers tend to be off center. So it's on this side of this chromosome pair. So these short pieces of chromosomes to this right side of the crossover become these short arms. So this folds up, that folds down. And then these long pieces of chromosome to the left of the crossover fold out this way and become the long arms. So the bivalent structure is this pair of homologous chromosomes linked by a crossover, and it looks like this cross-shaped structure. But then during the meiotic divisions, everything condenses down further. So the short arms kind of tuck in and they become very hard to see. And so during the meiotic divisions and all the images and all the diagrams I'll show you, the chromosomes look basically like this bilobed two blob structure. But you can think of it as the two homologous chromosomes, one on each side, and they're linked in the center by a crossover. And then in C. elegans, it's quite convenient. There's a small number of chromosomes, so it's easy to visualize them all. There's 6 of these homologous chromosome pairs, which means there will be 6 bivalents. And so now, quite a number of years ago, when I was a postdoc in Anne Villeneuve's lab, I discovered something really surprising about the way these bivalent chromosomes associated with spindles in C. elegans oocytes. And that's that they form predominantly lateral associations. So in the intro, I reminded you that usually in normal cell division, you have microtubules that embed into structures called kinetochores. And so they have these end-on attachments, as shown here. And in spermatocyte meiosis, so same organism, but in spermatocytes have centrosomes, so are kind of more normal spindles. You have end-on attachments. However, in the oocyte, the microtubule bundles predominantly run along the sides of the bivalent. These chromosomes do have kinetochores at their ends, but there aren't visible end-on attachments. And so just to kind of come back to this Bovary picture, it's a little hard to see on the projector, but if you actually look at this drawing from Bovary from the 1800s, you can get a sense that actually even in these drawings, there are also these lateral bundles of microtubule, what we now know are microtubules that run alongside these chromosomes. So it wasn't appreciated at the time, but this actually looks a lot like C. elegans today. And so when I saw this organization, I was really excited by it, but very confused. How are chromosomes moving in spindles if you have side attachments instead of these end-on attachments that can push and pull the chromosomes? And so I started looking for motor proteins that might be on the chromosomes, because the idea being maybe these are like train tracks, and you could put a motor protein, attach it to your chromosome, and that motor could walk the chromosome along the tracks. And so I found a motor protein that formed these beautiful ring-like structures right here at the center of the bivalent, as diagrammed here. And since then, we've also identified a bunch of other proteins in that same structure. So we now call it the ring complex, or RC. We now know, this was many years ago, so we now know that if we make genetic mutants where chromosomes lack ring complexes, they can't align on the spindle. And so that puts us with a model that these RCs, these ring complexes, are helping to power chromosome movement along these laterally associated microtubule bundles. And as I said, the first discovery of the ring complex, I found that it contained a kinesin called KLP19, which we think is helping to power the movement, but also AIR2, or Aurora B kinase, which is part of a chromosome passenger complex, or CPC. And I found that the CPC was required to target KLP19, the kinesin to the complex. And in the many years since, as other components have been discovered, we also know that the CPC is required to target those components. And so you don't have to memorize this list of proteins for the talks, but if you just look at the categories, there are kinases in the ring complex, there's microtubule binding proteins, there's motors. So it's a really interesting protein complex. And then if you look at the bottom of this list, you also might notice SUMO. So SUMO is a small protein that can get conjugated to other proteins as a post-translational modification. And so SUMO and its conjugating enzymes and one of its deconjugating enzymes are also in the ring complex. And so the RC is thought to be what's called a SUMO-SIM network. So some of the RC components are known to be SUMOlated to get SUMOs attached to them. And others have what are called SUMO interaction motifs, or SIMs. So those bind to SUMOs. So the prevailing model for how the ring complex assembles is that SUMOlated proteins then have SUMOs, and then proteins with SUMO interacting motifs bind to those SUMOs, and then some of them get SUMOlated, and then the complex is built as a network. And so kind of with this background then, my lab really focuses in a couple of different areas, but all related to this unique form of cell division. We're interested in understanding how the ring complex is built and organized, how this list of ring complex components contributes to oocyte meiosis. And key to the lab from the beginning has been this question of how the acentrosomal spindles form and how they're stabilized. So to show you some of kind of the early work on how these spindles form, just to orient you to the pathway, one of the first papers from my lab documented stages of spindle assembly in oocytes in C. elegans. So this is what you're looking at is a strain where tubulin, the microtubules, are marked with GFP, and histones are also marked with GFP. And so you're seeing chromosomes and microtubules both in the same color. And this is what you're watching the spindles form. So right here, what we're looking at is an intact nuclear envelope. This haze is trapped GFP histone molecules within the intact nucleus. And these are the 6 bivalent structures. And then as the nuclear envelope starts to break down, microtubules are nucleated first just interior to the disassembling nuclear envelope. The envelope isn't marked in these images, but we've stained for it in other images. And so there's membrane kind of outside of these microtubules, we think constraining the early microtubules into this, what we call a cage-like microtubule structure. And then microtubules start to reorganize. So microtubules are sorted so that the ends start to come together and form pole-like structures. And at first there's lots of these poles, but then the poles slowly coalesce together until you get a bipolar spindle. And so I mentioned that microtubules are polarized. There's plus and minus ends. So we were curious to know where the minus ends, where the plus ends are in these at these different stages. And so we stained with an antibody to a protein called ASPM1, which is a marker of microtubule minus ends. And so you'll see ASPM1 in some of my other images in this talk. Just think of it as a minus N marker. And so at this cage stage, we see ASPM1 dots kind of all over this cage structure. Can't really find a pattern. We think that microtubules, therefore, are nucleated fairly randomly. But then by the multipolar stage, these ASPM1 dots, the red dots, are enriched at the periphery of the structure, so away from the chromosomes. So microtubule minus ends are sorted outwards. And then by the time you get to a bipolar spindle, of course, minus ends should be at the poles, and all spindles we know of, so they're enriched at the poles. And so we have a model for acentrosomal spindle assembly that we've been building on over the years, where microtubules are nucleated randomly, then sorted by motor proteins so that the minus ends are enriched away from the chromosomes. Minus ends can then find each other and come together, and then these poles are coalesced until you finally get bipolar spindle. And so a lot of the early studies in my lab were trying to find proteins that act at various steps along this pathway, and that's something we continue to do. But in recent years, we've also been really interested in a related question, which is once you assemble the spindle, what factors are important to stabilize that structure? And the reason we got excited by that is because of a beautiful landmark study by another lab on human oocytes in 2015. And what they showed is that human oocyte spindles are very unstable. So they could film spindles form, they would form bipolar structures, but they have to sit around in metaphase 2 for a long time. And these spindles often reverted back to this multipolar stage. They would go multipolar, bipolar, and the poles would split apart frequently. And this created a large number of chromosome segregation errors. And so we've also been really interested in understanding what proteins on a molecular level are actually stabilizing the structure. And so to get at this question, around the same time, we started using a system called the auxin-inducible degradation system, which allows us to rapidly deplete proteins from cells so that we can let spindles form, remove a protein, and then see what happens. So this system was adapted for C. elegans by Abby Dernberg's lab, and it allows tissue-specific depletion of proteins on really short timescales. So what you do is you have your protein of interest, and you tag it with an AID tag or a Degron tag. And then we often add a GFP just so we can visualize it. And then in the worm, you also express a protein called TIR1, which is an E3 ubiquitin ligase. And the way the system works is that if you add a small molecule called auxin, this brings TIR1 to the AID tag, and then TIR1 can ubiquitinate the target protein and degrade it. And we've actually optimized this system for use in isolated oocytes, and we've shown that we can deplete proteins in minutes. And so, as I mentioned, this allows us to not only probe requirements in spindle assembly if we deplete a protein before the spindle forms, like we could before with RNAi, but now we can also probe requirements in maintaining spindle integrity. Because if you can get rid of something in minutes, you can let a spindle form and then watch what happens when you remove it. So this was the first published paper we had using the AID system in this way. And it doesn't matter for this talk what this protein is, but this is a protein called ZIG9, which localizes across the spindle. So this is our Degron tagged ZIG9, tagged with GFP. And then we have tubulin over here in a different color. And so we dissect the oocyte into auxin and as quickly as possible, start filming. And so you'll notice that when the movie starts, this protein rapidly goes away. It's usually gone by about 5 minutes. And then we can watch what happens to the spindle. And so this spindle becomes extremely unstable. Poles start splitting apart. I'll play that one more time. So when the spindle starts, it looks like a nice solid football structure. And then the poles become highly unstable. And so we've now applied this approach in multiple papers with different proteins. This is a protein called ZIG8. Now you're actually not looking at ZIG8, you're just looking at the tubulin and the chromosomes. But when we add auxin, this slightly different phenotype, the spindle gets very long. The center of the spindle becomes very disorganized. And then this is depleting dynein. Here that you're looking at GFP tubulin, GFP histone. And in this one, the poles become very wide. So they start off really pointy and they become very wide. So over the years, we've been using this system now to probe requirements for various conserved proteins in spindle assembly and stability. And so for the rest of the talk, then I'm going to tell you two short stories, both of which involve using the AID system to ask different questions related to oocyte meiosis. First, I'll tell you about some new roles we found for conserved mitotic kinase in oocytes. And second, I'll tell you an unpublished story we're just now developing, looking for a role of the ring complex in promoting spindle organization. So I'll start with this first one. So this story focuses on a protein called PLK1. So PLK1 is a conserved, highly essential mitotic kinase called polo-like kinase 1. Plays many important roles in mitosis, but it had been shown by multiple labs that if you deplete PLK1 by RNAi in C. elegans, then the nuclear envelope does not break down. So it was well known that PLK1 was required for nuclear envelope breakdown. So for many years, we didn't have a way of asking whether PLK1 also played a role in spindle assembly, because if we depleted the protein and the nuclear envelope doesn't break down, we can't look at what happens to spindle assembly. But with the AID system, once we got that working well in the lab, we decided to start looking at this protein. So our goal was to use it to probe roles for PLK1 after nuclear envelope breakdown had already occurred. And so first, we made our Degron strain where we tagged PLK1 with GFP and with an AID tag. So this is actually imaging of PLK1 using a GFP antibody. You can see PLK1 is in multiple locations. So here's the spindle poles here and here. It's also on the chromosomes. And if you look at these little lines, those are the ring complexes in the center. And so we can see it. These strains are viable. And so we wanted to see if the AID system could work to degrade PLK1. So first, we just wanted to validate that we were getting a robust depletion. And so we put the PLK1 AID worms on auxin for 18 hours. Like we put auxin in the plates, let it soak into the worms, and have a long-term depletion that kind of mimicked RNAi. And in those cases, we saw the published phenotype of defective nuclear envelope breakdown. So what you're looking at in these images, often I'll have the full embryo over on the left, and then a zoom of a specific region over on the right. And so in a normal embryo, you have a spindle forming, the nuclear envelope has broken down. This is the oocyte spindle. But here, when we degrade PLK1, you have this hollow region of tubulin, so no tubulin inside, and you have a nuclear envelope marker, EMR1. And so great, this strain is working for a robust PLK1 depletion. And now we wanted to ask what happens if we now instead deplete PLK1 after spindles assemble. And so this is our kind of assay where we dissect oocytes into auxin. So first our control. So in these movies, you're actually looking at GFP tubulin and GFP PLK1 is tagged. So it's kind of on the spindle and the chromosomes, and M-cherry histone. And so without auxin, over the time course of our experiments, spindles remain bipolar. Chromosomes are kind of oscillating around. We can image spindles, they're quite stable. But when we add auxin to deplete PLK1, the spindle kind of just almost melts apart. So the poles remain intact. They still are pointy. Play that one more time. But the center of the spindle just completely collapses. So dramatic spindle defects when we deplete PLK1. So we decided to perform immunofluorescence microscopy to better characterize what these defective structures look like. So here on the left is minus auxin. Again, now we're looking at DNA tubulin, and we stained for the minus end marker, ASPM1. So again, ASPM1 is normally at spindle poles. And as expected, since we knew we were seeing dramatic spindle defects, when we looked at the spindle, you can see ASPM1 and microtubules are completely disorganized all over the spindle. We can quantify that and really just look at this pink bar. This is the disorganized category. We see mostly disorganized structures. But in doing this immunofluorescence, we actually noticed something else that was interesting and that we didn't expect. And that's that we saw a lot of this red ASPM1 staining not right by the spindle, instead in other clusters of microtubules in other locations. So if we now take that same image, but now we're zooming out to look at the whole embryo. So again, in a wild type embryo, here's the oocyte spindle. If you look at ASPM1, it's at the poles. You don't see a lot of ASPM1 or super concentrated areas of tubulin in other regions of the oocyte. But here, with PLK1 depletion, here's the collapsed spindle, but we're also seeing a bunch of ASPM1 foci with associated microtubules, suggesting that PLK1 depletion is causing formation of microtubule asters, other parts of the cell. And so this is just a depletion experiment where we're kind of now looking at the whole cell instead of just the spindle collapsing. So these are unarrested, so they're actually progressing through the meiotic divisions. I forgot to tell you, but the other movies I told you, we had induced an artificial metaphase arrest so we could keep the oocytes stable for a long time. But these control oocytes progress normally. But now with auxin, the spindle is collapsed, but you'll start to see asters of microtubules forming in other parts of the embryo and kind of coalescing together. I think that's looped, yeah. So just look in the center of the embryo here. You'll see extra microtubules assembling, and then often, probably with cytoplasmic flows, they collect together. So our conclusion from this is that PLK1, in addition to being important for spindle assembly and stability, is also appears to be suppressing microtubule nucleation at other parts of the oocyte in other regions. And interestingly, when we were analyzing these images even further, we noticed something else puzzling. And that's that often we would see clusters of microtubules and ASPM1 staining near the sperm DNA. So as I mentioned before, here's the oocyte spindle, but in this system, the sperm has fertilized the oocyte at this stage, and a lot of microtubule density was surrounding the sperm. So if we quantified it, we see a large amount of microtubules in this region. And so this was puzzling because, as I told you, usually the sperm is present in the egg while the oocyte divisions are happening, and it has associated centrioles. However, if you look in a control oocyte, there are no concentrated regions of microtubules near this sperm. So these sperm-associated centrioles have not matured into active centrosomes at this stage. They're not nucleating microtubules. But in the PLK1 depletion, we saw a ton of microtubule intensity in this region. And so this is just the same thing in diagram form. So here's the C. elegans germline. It's like a production line where oocytes are being produced, from left to right. They're maturing, and then they're going to get fertilized by sperm, and then that's what triggers the meiotic divisions to happen. And so again, the centrioles are eliminated during oogenesis. Oocytes lack their own centrioles, but then the sperm brings in a centriole pair upon fertilization. But it's not active to nucleate microtubules at this stage. It's only during mitosis that then these centrioles mature into centrosomes that nucleate microtubules. So during mitosis, centrosomes become competent for microtubule nucleation. And so this is just a diagram from this Woodruff paper showing a model for how centrosome maturation occurs in C. elegans. So again, these centrioles are recruiting proteins that are going to become the pericentriolar material, or PCM. And one of these proteins is called SPUD5 or CDK-RAP2, and that is thought to oligomerize and form a scaffold that then can recruit other PCM components, including the components that are going to nucleate the microtubules. And it was already known from studies from Frank McNally's lab that in the oocyte, this SPUD5 protein is not normally recruited to sperm centrioles until after the meiotic divisions, the oocyte spindle divisions are complete. But because we were seeing premature, microtubule nucleation near the sperm, we wanted to ask whether PLK1 depletion was perhaps causing premature centrosome maturation in the oocyte. And so we stained for SPUD5. So what you're looking at here, I've been showing you lots of zooms of the spindle region. This is now a zoom of the sperm DNA region. So here's the sperm DNA in a control minus auxin embryo. And if you look down here at SPUD5, you don't see really, there's background staining, but you don't see any concentrations. But upon adding auxin to deplete PLK1, you now see foci of SPUD5 in addition to higher microtubule density. And if you look at the quantification, we would either see one focus of SPUD5 or two foci of SPUD5, never three. And so our assumption is that these foci represent the centrioles. So, the sperm brings in a centriole pair. Usually they're tightly coupled and close together. In those cases, you probably can't resolve them, so you see one. But if they've moved apart so that you can distinguish them, then you see SPUD5 on both. And we also looked at other PCM components. This is one called TAC1 that's recruited to the centrosomes, and that was also recruited to the centrioles. And another interesting phenotype is that we also noticed that in a lot of cases, when we had these phenotypes, the sperm and the oocyte spindle moved closer together. So usually the sperm is at one end of the embryo and the oocyte spindle is near the cortex at the very other end. But in these PLK1 depleted situation, these moved closer together. So we quantified the center of the sperm here to the center of the oocyte chromosomes, and these distances were decreased. And so the idea is that by preventing premature centrosome maturation, and you're preventing excess microtubule polymerization that's going to interfere with spindle and sperm positioning. And so PLK1 normally, by preventing this, is allowing the meiotic divisions to be completed without interference. By that, I mean, without the sperm coming all the way over to the oocyte chromosomes and getting in the way, because sometimes the distances were really close together. So how is PLK1 performing these roles? Well, first, we were curious if it actually localized in this region of the cell, because no one had thought to look near the sperm. So again, this is the sperm DNA region. Here's the DNA. We just co-localized PLK1 with we just had an antibody from another lab to ZYG1. ZYG1 is an important mitotic kinase, but importantly for this talk, it's localized to the centriole. And so we can see PLK1 co-localizing with ZYG1, suggesting that PLK1 actually is present at the centriole where this is happening. Okay, so this is all really cool. It's an interesting phenotype, but the reason why all of this was really surprising to us is that PLK1 was known in mitosis to play exactly the opposite role from what I just told you. Okay, so in mitosis, this is a mitotic one cell stage embryo. Here's PLK1. It was shown by many labs in beautiful work, to be localized to the centrosomes, to the centrioles, but it has been very clearly demonstrated that PLK1 phosphorylates SPUD5 in mitosis to promote SPUD5 oligomerization and centrosome maturation. So in mitosis, if you deplete PLK1, you don't get centrosome maturation. But in meiosis, in our system, when you deplete PLK1, we're getting premature centrosome maturation, which was really confusing, but actually really interesting, suggesting that this protein plays different roles in different contexts. And so finally, we wanted to know how does PLK1 prevent centrosome maturation in oocytes? So we still haven't fully answered this question, but we've started to try to address some potential mechanisms. And the first protein we decided to look at was a protein called KCA1, which is an adapter for kinesin 1. So KCA stands for kinesin cargo adapter 1. The reason we wanted to look at these proteins is because there was a paper from Frank McNally's lab back in 2012 where they published that depletion of these proteins gave a very similar phenotype to what we're seeing for PLK1. So this is a figure from their paper. So on the top is wild type, on the bottom is KCA1 RNAi. So what you're looking at here is the sperm DNA with the arrow. And if you notice, if you look over here, they're standing for SPUD5. When you deplete KCA1, SPUD5 localizes to foci near the sperm DNA, and you get premature microtubule nucleation. And they actually showed these shortened sperm spindle differences as well, which is one of the reasons we thought to look for it in our phenotype. So same phenotype phenotype here when you deplete KCA1. And so we wanted to start to look at whether PLK1 depletion might be impacting KCA1 in any way. And so the way we decided to look at that was just simply looking at KCA1 localization. So we got an antibody from the McNally lab. So actually in their paper, they also said that it forms, it's very diffusely localized near the sperm DNA. So again, this is the sperm DNA region here. Here's KCA1. It doesn't look like much of anything. They described it as like a diffuse shell of KCA1 around the sperm DNA, and we saw something kind of similar. But when we depleted PLK1, we noticed now KCA1 was formed very strong, distinct foci, suggesting that maybe in the absence of PLK1, it was being recruited to the sperm centrioles. Suggesting that PLK1 might play some role in localizing KCA1 and therefore regulating its function. Although I will say we still don't know how KCA1 and kinesin 1 might be impacting, microtubule nucleation. Okay, so finally for this story, we decided to look at whether the kinase activity of PLK1 was required for all of these new functions. So as I mentioned, PLK1 is a really important mitotic kinase. We're assuming it's playing these roles by phosphorylating proteins, but we wanted to look. And so we decided to use something called an analog sensitive allele. So what this is that you actually take your kinase of interest and you can make specific mutations in the kinase domain, the ATP binding pocket of your kinase, to kind of open up a little pocket to allow it to bind an analog of ATP that's bulkier. So this is your analog sensitive allele. It has this extra little domain cut out of it, allowing it to bind this bulky ATP analog. Normal kinases can't bind these bulky analogs. They just bind ATP. And so if you add this bulky analog, it's not going to inhibit any normal kinases in your cell, but it will inhibit whatever kinase you've added these mutations to. And lucky for us, the UW-Madison labs had already made an analog sensitive PLK1 mutant. So they mutated, 2 amino acids to create this bulky ATP binding pocket. And then this will accommodate binding of these bulky alleles. So we decided to use this strain to see if PLK1's kinase activity was required for its roles in oocytes. And long story short, we found that it was. So basically every phenotype we saw with PLK1 depletion, we also saw after 10 minutes of soaking of this analog. So this is the NMPP1 here. So looking around the sperm DNA, we saw extra microtubules. We also saw SPD5 localization. So here's SPD5 in foci. We saw increased tubulin intensity near sperm. We saw the spindle orientation defects, et cetera. So all of these functions that we found require kinase activity. And so conclusions for this part of the talk is that PLK1 is playing, new roles in oocytes. So in normal spindles, the spindle is forming and the sperm centrioles are present but are, off. When we deplete PLK1, first we see spindle defects. We also see ectopic microtubule asters throughout the cytoplasm. We also see PCM recruitment to the sperm centrioles, and that ultimately results in microtubule nucleation from this now sperm centrosome prematurely. And really the thing that was the most cool about this story is that genetics led us to a really unexpected function, and that PLK1 plays an opposite role in oocytes to its canonical role in mitosis. Okay, so in the last few minutes, I'm going to tell you a shorter story on some unpublished work, just to give you a flavor of the types of questions we're asking in the other part of the lab, focusing on chromosomes and the ring complex. And so back to the intro, this is basically a slide I've already shown you. Remember that there's a ring complex composed of a lot of proteins that forms around the center of the bivalent, and then you have lateral microtubule associations. And we know that if we create chromosomes lacking ring complexes, they fail to align, that the CPC or Aurora B is required to assemble the ring complex. And also, I didn't tell you before, but in a 2021 paper, we found that depleting AIR2 to prevent ring complex assembly also results in massive defects in spindle assembly. And so we decided to ask, could the RC have a role in promoting normal spindle organization? And specifically, this really unusual lateral bundle configuration that is present in oocytes, but not to our knowledge in normal cells. And so the way we decided to look at this is to look to see if lateral bundles were able to form around chromosomes lacking ring complexes. So we used a genetic mutant from Abby Dernberg's lab, in HIM8. HIM8 is a protein that mediates pairing of the X chromosomes. So in a HIM8 mutant, you have two unpaired homologs or univalents. These do not form ring complexes. So you have 5 bivalents with ring complexes and 2 univalents without. So that's shown here. Here are the 2 univalents with the arrows, and you can see there's no ring complexes. So do these chromosomes assemble lateral bundles? So in order to visualize them, we had to use a trick, and that is to actually perform RNAi of an essential kinesin to form monopolar spindles. This is because in bipolar spindles, the chromosomes are so close together at the metaphase plate, and there's so many microtubules, it's impossible to kind of see the organization. But when we spread them out, we can get a good look at them. And so what you're looking at here is the same monopolar spindle, but they're 2 different Z slices. So the Z slice in these 2 images on top is focused around this chromosome with the ring complex. You can see these strong microtubules along the side. And this on the bottom is focused around this little univalent with no ring complex. And there are the microtubules. So quantifying fluorescence intensity and line scans across a bunch of bivalents with ring complexes and univalents without, you can see really strong peaks in the green of microtubule intensity along the sides of chromosomes with ring complexes. So the magenta here is AIR2, one of those ring complex components. So here's the ring complex and here are the peaks. And we see some microtubules around univalents, but nowhere near the same intensity. So that was cool. But we also just wondered, well, maybe this is happening because bivalents are twice the size, there's twice the amount of DNA. Maybe that's why they're able to assemble lateral bundles, not because of the ring complex. So we wanted to have a way of looking at the same size chromosomes, but some had rings and some didn't. And so we took advantage of another genetic mutant called SPO11. So SPO11 creates double-strand breaks. So in a SPO11 mutant, you cannot get the homologs to recombine, and you end up with 12 univalents. But interestingly, we found that only some of the 12 univalents loaded ring complexes and some did not. And so that's shown here. So this is a SPO11 mutant. These are all univalents. And if you look at AIR2, and this is SUMO, a downstream component of AIR2, you can see that they're not on all the chromosomes. And so we quantified this. And so in SPO11 mutant spindles, there's always 12 univalents, but a variable number of ring complexes are able to load. So that's really cool. We wanted to use this then to compare microtubule organization around these different types of chromosomes. And so we use the same monopolar spindle trick. So here is a Z slice focused around this univalent that has a ring complex. And you see strong microtubules along the side. Here's the ring complex. And here are 2 univalents at different places. There are microtubules around because there's a lot of microtubules in the structure, but it's kind of more of a haze instead of 2 prominent bundles along the side. We've also done the line scans on these, and they look similar. So finally then, we have a hypothesis that these ring complexes are maybe somehow helping to assemble these lateral bundles. Our prediction then is that if we were able to remove ring complexes from pre-assembled chromosomes, this should disrupt spindle architecture. And as I told you, we knew that AIR2 was required to build the RC. So we decided to see if we could use acute AIR2 depletion to disassemble the RC. So let it form and then remove it. And these are just proof of principle that we can remove ring complexes from chromosomes. So this is an AIR2 AID strain. Here's AIR2 and SUMO on normal bivalents with no auxin. They form a big peak between the 2 lobes of the chromosome. And this is a short auxin incubation. AIR2 is gone because that's what we're depleting. But then SUMO is either delocalized or missing. And so here's the 2 lobes of the bivalent in a line scan. And you see AIR2 and SUMO are basically gone. So then when so we can now acutely remove ring complexes from chromosomes with a short auxin incubation. We next then wanted to ask if microtubules are then destabilized. And so first we did immunofluorescence. So on the top is no auxin, on the bottom is with auxin, and then we can quantify fluorescence intensity and see that general microtubule intensity goes down when we acutely deplete AIR2. And then we can then use a strain with AIR2 tagged with GFP and tubulin tagged with GFP to visualize the depletion of AIR2 on the spindle. And so this is just the control. So you see tubulin and AIR2. This is just control. So the spindles maintain their integrity. You can see AIR2 in these little ring complexes going around the spindle. And then when we dissect into auxin, what we see is you'll start to see those ring complexes go away. You don't see those strong lines anymore, and the spindle collapses. So conclusions for this part, this is a story in progress, but we know that chromosomes without RCs, the ring complex, fail to stabilize these lateral bundles. They look more like this, that Aurora B kinase is required to maintain RC integrity, and that RC removal is resulting in disrupted spindle architecture. And so we're trying to wrap up this story, but we do have a few outstanding questions we do want to address first. The first of which is we don't actually know if the stabilization is the ring complex itself, like you need all of those components there, or if it's just the CPC and Aurora B that are providing the stabilization effect. So we're trying to tease that apart. And second, we don't know, we keep calling it the ring complex. We don't know if the ring part of it is important. Do you need these proteins organized into a ring structure, or is it just concentrating it on the chromosomes enough? And so to try to answer this, we've been playing around with this new system developed by Abby Dernberg's lab and published just in 2024, where they've now are repurposing the AID system to perform something called CIP, or chemically induced proximity. So the idea behind this is pretty cool. So you can repurpose existing AID strains you have, where you have a protein of interest tagged with a degron tag. But instead of expressing the tier 1 E3 ligase in order to degrade a protein, you instead fuse a mutated form of tier 1 to a second protein of interest. So it's mutated, it has a few amino acid changes, so it can no longer function as an E3 ligase. But the idea is that when you add auxin, it still brings your mutated tier 1 to your AID tag, which then can bring your protein of interest in proximity with a second protein of interest, driving potentially relocalization in the cell. So we've been building strains where we're fusing key meiotic proteins in different locations in the cell with this tier 1 CIP, and we now have these 4 strains working and validated. So we have an ASPM1 version. This is a spindle pole protein, one of the ring complex components. We have HCP4, which is a broad chromosomal localization, and then a kinetochore protein. And so currently we don't have data for this yet, but the idea is to cross our AIR2 AID strain with this one where it's on the chromosomes but not in the ring, and then add auxin to try to relocalize AIR2 away from the ring to ask if the ring organization is important, or if you just need AIR2 activity on the chromosome somewhere for the lateral bundles to form. And just, this has nothing to do with that question, but this is our first proof of concept that this can work. This was one of the first ones we got working. So what you're looking at here is the ASPM1 tier 1 CIP fusion. So this is a pole protein. And this is an AID tag protein called SPUD5, which is actually, it's hard to tell here, but it's on the spindle, but like just above background and quite diffuse. And when we put it with this ASPM1 tier 1 CIP and add auxin, we now see SPUD5 strongly relocalized to the poles, suggesting that we can move things around in these cells. So this is kind of one place where I hope my lab is going, is now being able to use these AID strains we've been working with in order to not only deplete things, but to move them around to look for location-specific functions. So that's kind of it. So altogether, I hope I've convinced you that oocyte meiosis is a really cool form of cell division, that there's lots of interesting questions, and we're continuing to try to figure out how this works. So with that, I'd like to thank my lab. So the people listed in red are people that contributed to the work I talked about today. So the PLK1 story was the work of a fantastic former student who recently graduated, Juhi Narula. Jordi Martinez has been driving the AIR2 ring complex project. And then the SIP system in the lab was developed by Emily and Lizzie. So with that, I'd be happy to take questions.
Yeah.
Do you have any thoughts about what the substrate of the kinase PLK1 is in the system?
I bet there are a lot of substrates. So I didn't show you this because it was all negative data, but we did spend some months like kind of looking for like candidate target proteins like KCA1 and kinesin-1. And we had another candidate I didn't tell you about that we thought might be generating the asters that were not sperm associated. We looked for consensus sites and then mutated them to see if it recapitulated the phenotypes. And so far we haven't been successful in that, but it's possible that PLK1 is also phosphorylating multiple domains. So the negative data doesn't really tell us anything. But yeah, if you just look through the literature, PLK1 is known to phosphorylate lots of things. So it's probably modulating these different phenotypes through different targets. There's probably some spindle targets, some sperm targets, that kind of thing. But great question. We would love to go more in that direction.
I was just thinking with your modified ATP binding pocket,
Yeah.
you could identify the substrates by using an ATP analog that the phosphate is linked to some molecule that can be tagged.
Yeah, we have thought about, that's a great idea. We have thought about that, but we definitely haven't started work on it yet. But yeah, we would, it is possible, with C. elegans, you probably know, but to grow large quantities of worms and do pull downs and this kind of thing and mass spec. So certainly that's a great idea. Thank you.
Really lovely talk.
Thanks.
Super cool stuff.
Oh, thanks.
My question is related to the kind of coalescence of the microtubules without a central.
Yeah.
Really, I'm just curious if you thought about if there's something there that's actually helping them kind of maintain that bipolar shape.
Yeah, that's a fantastic question. So we have thought a lot about that and we don't know yet. So it kind of gets to a more philosophical question, but what is even a pole when you don't have a centrosome? So in mitosis, the pole is the centrosome. It's kind of defined by it. In meiosis, we don't know if there's like a substructure and a kind of stabilized thing that's drawing things together, or if it's just like cross-linked microtubules and binding proteins and they find each other. I will say that in mouse oocytes, another lab did some beautiful work showing that there is like a phase-separated compartment at the pole. They call it the LISD. It's the liquid-like spindle domain that has some proteins in it at the pole. Although when they disrupt the LISD, the poles still form. The spindles are less robust, but the poles still form. We haven't observed anything like that in C. elegans. None of the proteins, look like they could be forming like a separate compartment. They all just look kind of microtubule associated, but near the pole. But that we definitely would love to know that. And we've been doing some FRAP experiments to like look at the turnover of proteins at poles to try to see if we can identify a stabilized subpopulation of proteins at the poles. Most things do turn over that we found, although some turn over quite slowly and some fast. So we are trying to start teasing that apart, but it's kind of early days, but great question.
So right, there's defects in meiosis increase as oocyte age, and I was wondering if you'd use like a cog2 worm or something to look at kind of how these factors localize.
That's a great question. We haven't done that at all. But I get questions about that. So I really need to start like planning to do those experiments. As you said, like it's a really important question because in human oocytes, aging increases errors a lot. And our error rate in these oocytes is very low but they're young oocytes, in pretty young worms that we're looking at. Older worms, though, I will say will still generate, younger oocytes because of the production line nature. So it's in the human and mammalian systems, you generate an oocyte and then it sits arrested for many years at one stage. And in the worm, even if the worm's older, the oocytes are being continually produced. So I don't know if it would recapitulate the phenotypes, but it would be really cool to look. Definitely should look, yeah.
Yeah. What about anaphase? I assume they're defective when you knock down PLK1.
Yes.
And then have you thought about like just the fact that metaphase is so prolonged now, if that's why you're seeing those numbers?
Yeah, that's a great question. So interestingly, in those live imaging experiments, when we deplete PLK1, they don't go into anaphase. So there aren't thought to be like checkpoints that arrest the cell cycle because of spindle defects in the system. So we don't know if PLK1 is also regulating the cell cycle and maybe APC activation or something. So we haven't been specifically able to look at anaphase. Although I will say that occasionally, because the way you do these experiments, you dissect, oocytes into auxin, and you go as fast as you can to find a normal looking spindle where nothing's depleted, and then you film it. And occasionally my student did find one that looked normal but immediately initiated anaphase. So like anaphase was proceeding as it was depleting, so we were able to kind of bypass, and there were some defects, but the numbers are really small because you have to get really lucky to catch that. So, but yes, I would certainly expect, because you're affecting the spindle and the middle of the spindle is falling apart and then everything collapses, that anaphase has got to be defective. So yeah, but it's really hard to catch because of that cell cycle arrest. Awesome.
So, one of the, I'm not very familiar in RC, but the RC complex, one of the list you show is very clear about including the condension. So let's
Yeah.
whether it's kinase and like microchip related or something in a kind of domain.
Yeah.
It's only one that proteins are kind of more for in DNA from inside. So
Yeah.
maybe set that head for something in a condensation or something in a binding between there or what?
Yeah, so it's CAPG1. That's good. Yeah, really insightful to notice that. So yeah, there's one condensin component, and probably that whole condensin complex is just that we only had the antibody to one, does localize in that domain. We haven't done a ton of work on the condensin, but yeah, it seems like condensin forms a specialized domain at, as you said, the DNA level that probably is helping to kind of build, from the DNA up. And I talked about how this complex is a sumo-sim network with certain things simulated and then recruiting other things. We do think there are proteins that are more internal to the DNA that bind directly to the DNA first. So there are a couple of CPC components that also bind DNA. And I think condensin is one of those. It's probably on like an inner layer that help, as part of the like start of the complex. But yeah, we haven't actually done a lot of work on condensin, which is, but it's a really interesting question. There's been a question down at the front, so I should, yeah, go ahead.
Well, real quick, logistically, how prohibitive is AID in terms of like both like labor, in terms of costs, in terms of how many proteins you could screen or look at?
Yeah, I mean, it's definitely like RNAi is like super easy. You can do screens of hundreds of proteins. This, you have to generate a strain. So I guess it depends on how efficient, you have CRISPR working in the lab. So it's a 44 amino acid tag for the degron. That is actually pretty like not hard to insert. For these when we add GFP, doing larger insertions, like we have to, it takes longer. It's harder to generate those strains. So, I mean, we started actually, ironically, we started making a lot of these strains in the pandemic when my lab was shut down because my students were all sitting at home, and there's a company called SUNY Biotech that creates strains, and it's expensive, but I said, throw some money at the problem. Maybe we can have some strains waiting in the lab when they, so you can order them, but it's money, or you can generate them. But we have, I think, about 15 or 20 AID strains in the lab that my lab is not huge, it's grad students and undergrads, but yeah, it's definitely possible on a handful of proteins in a short time frame. If you want to do it, screening is probably not the way to go. But if you have a strong candidate, it's definitely worth trying. Yeah. Yeah, sure.
So you invoke a lot of diagrams from researchers looking at meiosis in the 1800s.
Yeah.
Do you know how they even did that? Because I can't imagine doing that with the kind of technology they had back then.
Yeah, I mean, it is quite amazing that those early microscopes, these are all hand-drawn images from very early microscopy. And there are these, if you look through those books, you see like pages and pages of like drawn, cytokinesis and spindles and centrosomes. Bovary discovered the centrosome based on these diagrams. So very early light microscopy, they picked cell types that were larger. They picked, transparent samples. So, but yeah, it is quite amazing that they look almost like our spindles we capture with our microscopes today.
Can I ask one last question?
Oh, yeah.
Do you think maybe ARC-2/3 or some actin would be in the ring complex, because the Meyer lab pulled out a couple ARPs in the cohesion IP paper. Right?
Yeah, actually, we've never looked, and that's a great question. But there's quite a lot of literature now, especially in the mammalian field. So like mammalian mouse oocytes. A lot of labs have looked at spindle actin. You know, there's a lab. It came out of the shoe lab at Binion Mogacy, has started his lab at Yale now studying that. So within the mammalian spindle, and I think also Xanthopus, there is thought to be actin within these spindles. Whether it's in the ring complex, I don't know, but we should really look. We haven't looked yet. Yeah.
Right. Thanks, Sadie.
Thank you.
I will.